This article, titled Quantitative Proteomics Analysis of Cerebrospinal Fluid Reveals Putative Protein Biomarkers for Canine Non-Infectious Meningoencephalomyelitis, delves into the complexities of diagnosing and understanding non-infectious meningoencephalomyelitis, NIME, in dogs. This disease, characterized by inflammation in the brain and spinal cord, presents a significant diagnostic challenge for veterinarians due to its similarity to other neurological conditions. Accurately diagnosing NIME in dogs before death is difficult because of the overlapping clinical presentations, imaging findings, and cerebrospinal fluid, CSF, analysis results shared with other neurological diseases. The authors of this study aim to employ advanced quantitative proteomic techniques to uncover new biomarkers for NIME, potentially enabling earlier and more effective treatment. The study included 11 dogs, divided into two groups: a control group with 6 dogs diagnosed with intervertebral disc disease, IVDD, a common spinal cord problem, and a study group of 5 dogs diagnosed with NIME. The researchers chose dogs with IVDD as the control group instead of healthy dogs due to ethical concerns surrounding CSF collection, an invasive procedure, in animals without a medical need for it. In dogs with suspected IVDD, CSF collection is already part of the diagnostic process, making it an ethically justifiable choice. After a thorough clinical examination, blood tests, and imaging studies like computed tomography, CT, scans were performed on all dogs to confirm their diagnoses. Figure 1 provides a visual representation of the analytical workflow employed in the study. The figure outlines the steps taken to discover potential protein biomarkers in both CSF and serum samples from the dogs. The process started with sample collection from dogs with IVDD and NIME. Then, proteins in these samples were broken down using trypsin, an enzyme that breaks down proteins. The resulting fragments, known as peptides, were then meticulously analyzed using a technique called tandem mass spectrometry. This powerful technique helps identify and quantify the proteins present in the samples. The researchers collected CSF samples from all dogs during their diagnostic procedures, while serum samples were collected from a vein after the CSF collection. Tables 1 and 2 summarize essential information about the dogs included in the study. Table 1 focuses on the dogs diagnosed with IVDD, providing details about their breed, sex, age, the specific type and location of their disc herniation, the severity of spinal cord compression, clinical signs, and the medications they received before CSF collection. Table 2 provides a similar overview for the dogs diagnosed with NIME, including information on their breed, sex, age, the suspected type of NIME, findings from their CT scans and CSF analysis, observed clinical signs, and any treatments administered before CSF collection. The researchers discovered that the total protein concentration in the CSF of dogs with NIME was notably higher than in dogs with IVDD, as shown in Figure 2a. Further analysis using SDS page, a technique for separating proteins, revealed differences in the protein patterns between the two groups, Figure 2b. This finding indicated the presence of a unique set of proteins in the CSF of dogs with NIME. Figure 3 visually represents the findings from the Global Proteomics Survey conducted on the CSF samples. Figure 3a illustrates the number of protein groups identified in each group of dogs at a 1% false discovery rate, FDR. Figure 3b is a Venn diagram displaying the unique and shared proteins found in both groups of dogs. Lastly, Figure 3c showcases the results of a GO-term analysis, highlighting the biological processes associated with the proteins found exclusively in the NIME group. Using a sophisticated technique called label-free absolute quantification, the researchers were able to pinpoint 36 proteins in the CSF that showed significant differences in abundance between the two groups. These findings are visually represented in Figure 4. Figure 4a, a heat map, displays the proteins with significant differences in abundance between the two groups. Figure 4b, a principal component analysis, PCA, plot, shows how the samples cluster based on their protein profiles. Interestingly, several of the proteins found in higher amounts in the NIME group are known to play a role in the body's immune response and inflammation. These proteins could be key players in the development of NIME and potentially serve as diagnostic biomarkers. Conversely, many proteins found in lower amounts in the NIME group are involved in processes related to the development and function of the nervous system. This decrease suggests that NIME may disrupt these essential processes. Figure 5 presents box plots of the proteins exhibiting the most substantial changes in abundance between the two groups. Surprisingly, the analysis of serum samples did not reveal any significant differences between the two groups. The authors suggest that this could be because CSF, being in direct contact with the brain and spinal cord, reflects the changes happening in these areas more accurately than serum. The study's findings suggest that an overactive immune response and disrupted nerve cell function might be central to the development of NIME. This conclusion is based on the identified proteins that are either more or less abundant in dogs with NIME compared to those with IVDD. These insights could pave the way for new diagnostic tools and treatments for NIME in dogs. The researchers also point out that the proteins they identified might mirror those found in human autoimmune diseases, suggesting potential avenues for research and treatment development. However, the authors acknowledge that this study has limitations. The number of dogs included in the study was small, and the diagnosis of NIME was based on clinical signs and CT scan findings, which are not as definitive as examining tissue under a microscope. Additionally, the use of a control group consisting of dogs with a different neurological condition, IVDD, instead of healthy dogs, could have influenced the results. Therefore, further research with a larger number of dogs and a more diverse control group is needed to validate these findings and explore their implications fully. This study significantly advances our understanding of the intricate biological processes involved in NIME in dogs. The identified proteins could hold the key to developing more effective diagnostic tests and targeted treatment strategies for this challenging neurological disorder. From Vet NeuroJar, keep those minds inspired, hearts light, and tails wagging.